Well, hello, everyone. Welcome to the Malone Lab Chemical Hygiene Training from my couch. All right, let's see. What are we supposed to learn before we're working in a lab? We need to know that everyone is responsible for safety. Everybody must be trained. Everybody must be aware of all of the hazards associated with materials, and you must be able to handle chemicals in a safe manner. What does that mean? That means we're going to talk about different hazardous chemicals, specifically in our lab. You should have a general idea. And if you don't know how to deal with this specific chemical, you need to look it up or ask for help. What's the best way? Google it. That's pretty much the rules of the world now. If you don't know something, Google it and try to figure it out and move from there. Okay, so this is from the Chemical Hygiene Safety booklet at CSUN. And so here are some of the rules. I took out some little snippets of this giant book, so we all don't need to read it, and we're going to go through this. So everybody needs to do this stuff. Most of you will be research students. Some of you may be a teaching assistant, a student assistant, a volunteer, a technician, other supervisory faculty, right? You never know. So we need to figure out how we will work in the lab under the safety requirements established by each laboratory, by CSUN first, and then by my laboratory in general. All the training programs, so you need to take that lab safety test and anything else. We talk about how to use personal protective equipment. When should we use it? When do we not? And this means stuff for chemicals and normal lab work, not just COVID stuff. So obviously we will all be wearing masks and we will all be vaccinated, but that's not what this was originally referring to. Okay, so we have to make sure that we use it right and we keep it. Engineering controls, how do you use equipment? Everything needs to be done properly. Good chemical hygiene practices. What does that mean? Don't drink anything in the lab. Do not drink, nothing. For a while, somebody, I don't know if it was our building or if it was EH&S, was requiring us to put a sticker that says do not drink on every single bottle in the laboratory. Do not drink any liquids in the laboratory. Okay, no food or drink except in our little hallway, right, with the chairs. Can't cross that yellow line. So if the bottle set doesn't say do not drink, don't drink it anyways. It just makes sense. You're in a lab, don't drink stuff. Okay, so safety, standard operating procedures and other program documents, health and safety, just general stuff, right? I am the supervisor, all facts pertaining to incidents. If there was any injury or exposure, any actions, I need to know, okay? The other thing is if there's an injury, student health. If there's an accident, 911. Right, we'll go through that stuff. This is just the overview. All right, we need to know where is our chemical hygiene binder and our MSDS, material safety data sheets. Binders are near the front door to the left. So if you walk in the lab from the hallway, on your left, you will see this bench with all of these binders. Okay, that's this. It's going to be right to your left. Look up. They should be right there. So if we zoom in, MSDS, MSDS. Oh, here it is. Wow, sorry. Coming. This guy right there, chemical hygiene notebook. Okay, so if you cell phones are out, you can't Google something, all of the safety data sheets will be in one of these two folders here. One of the MSDSs and the chemical hygiene notebook tells you other information on what to do. Okay, so those are our main resources in the lab. Everybody needs to know that those are there right when you walk in the door on the left on the shelves. What else? Avoid working alone. Wear appropriate eye protection at all times. Normal glasses are not enough. So here's an example. Here we are over by gel electrophoresis, and this would be in the outer lab part on the right of the fume hood right here. This is part of the fume hood, right? So that would be here, right? With the sash and making a whole lot of noise. Flammable cabinets are under there. And this guy right here, that's the UV. Never, ever, ever turn that on without safety glasses. And better, which you really should always use, is a face mask protector. Okay, so let's zoom in there, right? That's a face shield. It's specifically for UV. It's not the cheapo face shields for COVID to keep people from snotting all over you. It has to be a real one, can't be that. Protects your eyes and your skin from UV. UV causes DNA damage. DNA damage results in mutations. Mutations result in cancer. Wear the head thing, okay? The other things over here might be ethidium bromide in a tray. We're going to talk about that as we go. This little handheld guy here, also a UV source. Everything over here, see how there's red lines and arrows? All of this is contaminated with ethidium bromide. Always, always, always wear gloves, okay? Pretty much everything in the lab, you need to wear gloves to touch. We try to keep everything clean, but you never know who's been touching what. Wear gloves, always in the lab. They're readily available. If most of them are the nitrile, non-latex, but you can check if you have some sort of latex allergy. Make sure you're not going to have a problem. Okay, what else? Okay, we need to know where are the flammable liquids. Okay. The only thing flammable that we should have are the ethanols and isopropanols. Okay, they'll be in a flammables cabinet. Chemical hazards, again, the MSDS. Protective equipment should be worn. We have lab coats available hanging on the wall. So if we walk in to the lab from the front door, right, here's that bookshelf that we looked at. If we turn right, we're coming down a hallways toward the refrigerators, lab coats on the wall. Okay, here's another view. This is from looking towards the front door. This is looking away from the front door. And so there's where you can always grab a lab coat that helps protect your clothing, helps protect your skin. Mostly we use that to protect us from bleach, so it doesn't wreck our clothes. Okay, so evacuation routes, spill cleanup, and waste disposal. All of those things are in manuals in the lab. If you need to know, which we do, you need to know where the fire extinguisher is. In our lab, it's in a cabinet on the wall in this hallway. Okay, we do not have a shower pull in our lab. We do have the eye wash in the sinks in the lab. There's a shower pull and an eye wash in the classrooms on our floor, but I'm not even sure if you don't have keys to those, that's not going to help you. So I will look into that. Our chemical spill kit should be in one of the rooms in the laboratory. We'll find that and make sure everybody knows where it is as well. What else? Aha, chemicals. Know how and where to properly store chemicals. So the only ones that we have to worry about are anything that's flammable or corrosive. They'll have a mark on the outside of the container that says flammable or corrosive. We have cabinets under the fume hood that say flammable or corrosive, and we need to separate those in there. We need to separate the acids and bases. That's all set up in the laboratory. You need to ask someone else if you don't know exactly what to do with those, or ask me, or don't touch it if you don't know what to do with it. So again, personal hygiene practices, that's using the personal protective equipment, the eyeglasses, the lab coat, closed-toed shoes. You don't want to spill anything. You don't want to drop anything sharp. Occasionally we use razor blades to extract bands out of gels from gel electrophoresis. So extracting DNA from gels, we use razor blades. So you need to be careful, right, with any sharp objects. Please don't use damaged glassware. If it's got a crack in the lid, then we need to put it in broken glassware. And we have to only use stuff for its designated purpose. So this right here is the other side of the shelves that in the front door. So right here is the front door. This is our office right there. And so if you look left, the top shelf on the other side is where the MSDS chemical hygiene stuff is. This is the opposite side. Lots of different solutions, nothing hazardous. Here, lots of different chemicals, nothing hazardous. They would be kept elsewhere. Always when you're dealing with any kind of chemical, eyeglasses, gloves, lab coat, face mask, which we'll have on anyways, because you don't want to inhale any particulates. Now, this is especially true if you are making LB agar. Yeast extract, which is one of the ingredients in LB agar, is extremely powdery. And when you weigh it, it goes poof. And the little particles go up into the air. And if you breathe it, it's disgusting and it makes you cough, right? That's not good. You don't want particles in your lungs. The other thing that's bad, particularly for your lungs, is SDS. SDS is a detergent. There's also SLS, so sodium lauryl sulfate. Also, both of those are detergents. They also are pretty powdery. Weighing those out, you don't want to inhale a detergent. What do detergents do? Break up lipids. What are our cell membranes made out of? Lipids. Not good. Wear a mask. You have to have one on for COVID. If that's ever over and we're not wearing masks, you're still wearing them in the lab, okay, especially around any chemicals. Moving on. Oh, here is our personal hygiene. Wash your hands. For all kinds of reasons. We keep our hand washed. Ooh, I just said don't inhale chemicals. No mouth pipetting. We have lots of the automatic pipetters. You don't have to use those stupid bulb things. We have lots of automatic mechanical pipetters. Never wash with solvents. What does that mean? Acetone, any of those nasty things. We don't even have those, but if you come across them. No eating, smoking, no putting on makeup, none of that stuff, except in our little hallway. Look, here's our little yellow line. So right there's our front door. This is looking at our fireplace. Virtual fireplace. And this is our food fridge. No chemicals are allowed in it. Food and drink only. You cannot cross the yellow line with food and drink. You can only have food and drink on this side. Nothing comes here. No makeup, no chewing gum, no nothing. No yellow line. Okay, this is the only place in the lab we're allowed to have that stuff. So you can leave water bottles, drinks, snacks, lunches, cabinets under here are also designated to store snacks and so forth. So you can look in there, see if there's anything to eat too. Yes, so this guy can't hold anything but food and drink. That's it. No chemicals, no lab stuff, no nothing. The rest of the refrigerators in the lab cannot have any food or drink in them. None, zero. We also have a microwave up here on this counter and coffee maker, I think, and maybe some drinking water. I can't remember what there is in. That's the only place you can have that stuff. No chemicals in that microwave. We have a microwave in the lab that's only for agarose and heating other chemicals. No food in that guy. It's all very segregated. Oh, look, here's the refrigerators I was talking about. So these are down the hallway. Walk in the door, turn right, look left, big freezer, refrigerator. There's another refrigerator in the tissue culture room. This is only for chemicals, right? There's gross stuff in here. Here's E. coli. Yummy. Let's put that in our lunch, right? Plates full of bacteria. Here, ooh, look at that. Do not drink. That's LB. That's for bacteria to drink. Yummy. right? More chemicals within here. Some of these could be a little bit caustic. There are chemicals within kits in very small amounts that aren't good for our skin. Don't get it on your skin. Wear gloves, okay? Down here, acrylamide, yuck, bad, neurotoxin. Don't get that on you. Wear gloves. Never, ever, ever drink anything in the lab from any refrigerator, freezer, or your own stuff. No, just don't do it and you'll be fine. What else, people? Oh my gosh. So I got to show all this stuff, right? Eye protection must be worn. We have safety goggles. We have the face mask for UV. Protective gloves. Yes, yes, we have those. We have gloves also for using anything cold going into the minus 80 degrees Celsius. That's really, really cold. Protect your hands. You can get frostbite right away. Liquid nitrogen. I don't know, that's like minus 280 degrees Celsius or some ridiculous. That just freeze your skin with one drop. Always wear protection. Never open liquid nitrogen tank without full protection, without a lab coat, without gloves, and without eye protection. Because if any of it pops up, it will blind you. That's just how it is. So don't do that. Safety glasses save your lives. Just like the poster in the chemistry rooms about Sandy or whoever didn't wear her safety glasses. Now she doesn't need them because she can't see. Okay. Confine your crazy mops, people. Don't wear big jewelry or anything like that that can get caught on stuff. It's not really a big deal, but you don't need to have, giant gold chains that might get stuck on stuff. Don't use defective equipment. That should not need to be said, but but I have to say it. We don't have disposable clothing. We don't have tearaway pants, unfortunately. But if we did, and gloves, you can't wear them in the hallway. You're not supposed to touch handles with your gloves, right? If you're wearing gloves, you get nasty stuff on them. That's why you're wearing them. So you can't go touch a handle where somebody might touch without a glove. Ew, no gloves on door handles, except interior in our lab. Anything interior in the lab, always wear gloves. Assume somebody touched it with something gross. Storage area for this equipment. There's safety glasses and goggles all over the place. I showed you where the lab coats are. There's a face shield near wherever we have the UV. And boxes of gloves show up here and there and everywhere. If you can't find the right size, we can always get those from the biology stockroom as well. All right, what else? Ooh, a minor chemical spill. Something you can safely deal with, right? There's no fires, explosions, terrible odors, right? You dumped a little bit of bleach on the counter. Wipe it up carefully. Put the stuff in the trash. A little alcohol. We use 70% ethanol to clean the counter. So, right, not a big deal. But if there's something, a bunch of ethidium bromide in liquid spills somewhere, let me know. I'll show you how to deal with it. But again, there's no possible fumes. There's none of that stuff. So you don't have to worry about it. We'll talk about the only things that you do have to worry about at the end of this little deal. Okay, but you got to tell people, whoa, sorry. Okay, alert people in the immediate area. Don't run away and not tell anybody. You're not going to get in trouble, right? Mistakes, accidents happen. You will get in trouble if you dump something, it catches on fire, and then you run away and don't tell anybody, right? The supervisor immediately. That would be me or any faculty nearby, or somebody that, if you're an undergrad, maybe a master's student, they can pass it on. We can always contact Wendy in the stockroom or anybody in the stockroom to come and help, or if you have a question about what you should try to do. Cleanup, we'll talk about the chemical spill container, and then make sure you are protected, or don't clean it up. Wait till you get protection. But tell somebody. All right. Uh-oh. Didn't change on here. Let me see. My screen share is not working. Oh, wow. I'm way behind. Okay, let's see. Let me stop share. Oh, mess up my tutorial. I am not doing this again. It took me all day just to get this file to the correct size and upload it and explain everything. And to this point of Zoom, I am not doing it again. Okay, so what did we miss? I was doing all this, not paying attention that you guys were looking at this. So I did this. See, it's all written. All this information is in the chemical hygiene booklet. If you want to go look at it, please do. Otherwise, you could read this. I kind of read it all to you, but it would have been really nice if it was actually on the screen while I was talking. So here you go. Yes, yes, I like you've done here. Yes. Pause the video if you need any longer. Ooh, spills. Again, talked about this. Pause the video. If you want to read through this, you need a little more time. Aha, now we're back. Biosafety level 1. Microorganisms not known to cause disease in healthy human adults. What is that? E. coli. All the lab strains are perfectly safe. So all the bacteria that we use to make DNA for us and make plasmids is perfectly safe. If you drank it, you'd be fine. But don't drink it. It's disgusting. Okay. Cell lines. All non-human cell lines are biosafety level 1. Human cell lines are between 1 and 2. So they're not technically level 2, but they're not level 1. Okay, so human cell lines would be like our HEC cells, our NIH, sorry, I'm lying, our HEC 293 T cells. Any other human B cells, human T cells, any human cells we use, we have to treat like biosafety, between biosafety level 1 and 2. And that means anything that touches human tissue or human cells must be disposed of in red bin. All pipettes, all flasks, all whatever, unless you bleach them for 24 hours in 10% bleach, then they can get thrown away in regular trash. So that's what we do, because creating the red, oh my gosh, it's not keeping up. The red, I swear I'm writing right down here, guys. The red bins cost lots of money to dispose because it's incinerated. It causes lots of pollution as well. So we will, as you're learning or using cell lines, these are the types of things that we will discuss. We have nothing biosafety level 3 or biosafety level 4, so we don't have to worry about that. Okay, I just changed slides and this didn't, so I'm going to stop it again. I'm not doing this again. I'm telling you people, I'm sorry. You can fast forward. Right? Hit me at 2.0 or whatever you guys do to speed on through. All right, biosafety levels 1 and 2. That's what we are here. So what you can see is our laminar flow hood. And that's in our tissue culture room. Laminar flow hood, it's open, right? And air blows at you. So it protects your culture from you, but not you from your culture. So as long as your culture isn't dangerous, like any non-human cell line and all our bacteria and stuff that we use, doesn't matter if the air is blowing at you. We don't want our germs and our contaminants in our cultures. If you have cell lines that are level 2, you need to use a different kind of laminar flow hood. Our human cells, since they are between, we're fine using them in this hood. We just need to dispose of everything in red. okay, except for flasks that we're going to bleach for 24 hours in the sink and then dump, rinse, and throw away like a normal garbage. The other deal we have is our fume hood. Our fume hood is not for growing cells. It is for any kind of nasty smelling stuff. Fumes, get it? Why it's called a fume hood. It sucks, it blows air up from the back and sucks in. So it kind of sucks everything up this way, okay? And so these aren't things in here that we're worried about getting our stuff on, because it's pulling it in. It's we don't want to smell anything bad. And there are a couple things that we do use in the fume hood, which we'll talk about. This guy here is our glass disposal. Any sharps, anything glass, okay? Not with human cell stuff on it, but anything that's just, you know, our regular biosafety level 1 stuff, but happens to be sharp. This includes yellow tips and blue tips, because they are considered sharps because they can poke through a garbage bag. We don't want to put those in the regular trash. We don't want those poking any of the custodians, even though nothing we use is dangerous. They don't know that. And so they need to go in glass. Also, broken glass goes in there, right? Razor blades, anything sharp, okay? Syringes always go in a red syringe specific box, okay? So that's the only thing that doesn't go, and we rarely use syringes. So wash your hands, no mouth pipetting, don't create the aerosols, right? The powdery stuff that we're going to breathe. Restrict access if somebody's working with something powdery, or make sure everybody's got their stuff on and they know what's going on. And last but not least, here is our stuff. Only things we need to worry about is ethidium bromide. Okay, this is, what is ethidium bromide? Ethidium bromide is a mutagen. That means we know it causes mutations in DNA and causes cancer. A carcinogen is something that's suspected of doing that. A mutagen is something we know does that. So no, ever touch this with bare hands. Always, always gloves. Be very careful. DMSO is a solvent we use for freezing and thawing cells. It's a neurotoxin. It absorbs through the skin. And don't drink it, okay? Both of the, so neurotoxin that absorbs through the skin is DMSO. We use it in very small amounts. Don't spill it on you. You have gloves on. You don't need to worry. Acrylamide, bisacrylamide are for polyacrylamide gel electrophoresis. These two guys are also neurotoxins, but you'd have to drink it in order for it to be a problem. Don't drink it. Timid helps cause polymerization. It stinks. Open only in hood, fume hood. If you really sniff it up, you might get a headache. It's not good for you. Same with DTT and BME. These stink. Yuck. Open in the fume hood only. I'm super, super sensitive to BME. It immediately gives me a headache. DTT smells super nasty, but I don't get a headache. I don't know. Only in the fume hood because they're gross. And if you're going to use BME for RNA extraction, I need to know, because I don't want to be in the lab at all, because you still have to bring stuff out to put it into centrifuges, et cetera, blah, blah, blah. Okay. DTT is fine. It stinks. Tell people you're working with so they can, you know, have a warning. What else? Bleach we use to decontaminate stuff, right? Bleach is caustic. Bleach is bad. You get it all. If you get it on you, it can burn if it's at a higher concentration, like out of the bleach jug. We use it at 10%, but still, it can still wreck your clothes and cause, you know, bleach marks, right? So be careful with that. Ethanol we use to decontaminate. It's also used in DNA and RNA precipitations. Isopropanol for DNA precipitation. These guys are flammable, right? We have them in the flammable cabinet. Why do we care? We're not smoking in the lab. because we might use Bunsen burners. Okay, somebody might be using a Bunsen burner and you're spilling ethanol, or you're cleaning the bench, not realizing they have a Bunsen burner on. Bunsen burners are used for sterilizing. We're picking colonies sometimes and some other things. We generally don't use it that often, but tell your lab mates or somebody else that's in the same room or sharing a bench, hey, I have the Bunsen burner on. Never leave a Bunsen burner on when you walk away. If you're walking to the other side of the lab, you turn it off. You don't get to leave it going. And that's another big thing. Don't have your hair down. It will burn in the Bunsen burner. You will forget and fling it around. So a braid or a bun or whatever, just don't get your hair on fire because it wrecks your hair. Okay. And that's pretty much it. All of these things you can Google to find out more and talk with the person training you. If it's not me, if it's one of the master's students, if it's Leslie, if it's another technician, if it's another student, ask questions. Bottom line, don't drink anything in the lab. Wear your safety goggles when you're using chemicals. Wear a lab coat if you're using bleach or you don't want to get stuff on your clothes. Closed-toed shoes, you don't want to drop a bunch of bleach or something nasty on your feet. You drop it on your shoes, they're going to stink. We take off your shoe and throw it in the hood. If it's your foot, then we can't cut off your foot and throw it in the hood. Okay, so just be careful, use common sense. The chemical hygiene booklet's up there. Any questions, we can Google, talk to me. We can also talk to environmental health and safety if you have any questions about anything regarding chemical hygiene. I thank you for listening, and I'll see you soon. Malone out. Okay, but now I have to find how to turn this off. So I'm not really out yet. Oh, I'm on the wrong thing. You'd think there'd just be a little button to stop recording, and there probably is, but I'm going to hit this. So now Malone out.